Today we're going to talk about experimental procedures. Let me tell you a little bit about how this little video is going to work. You have a document that looks like this that you can open through Schoology. It's number 11, Scientific Procedures. I will be taking you through all of this information, including the first example, in this PowerPoint. So you have two options. You can just listen to the whole video and then go back and fill in some of these notes, or of course you could watch the video and kind of go back and forth, pause the video, fill in something, pause the video, fill in something, that kind of idea. So what I want, just to make sure that you know kind of what's going on for today. So let's go ahead and get started. So again, if you have not already opened up 11 scientific procedures, go back and open that up, and then let's get ready to learn some stuff about experimental procedures. Here we go. All right, so we have been talking a lot about what science is. We know what it means to do science. We made an observation, we asked questions, we learned some content, and now we know some stuff about variables for experiments. So the thing we need to do next is tell other scientists or other people about the experiments that we have been doing. And that means we're going to learn some things about experimental procedures. And the reason this is good is we want scientists to be able to check our work and other scientists to be able to repeat our work. So what we want to focus on today is this question: what makes a good scientific procedure? Well, the first thing is that it tells the scientist what the scientist did in steps that make sense for another scientist to follow. Repeatability is important, so if I did an experiment, I want this person to also be able to do that same experiment and get pretty similar results, because if no other people can get the same results I did, that would be bad, because no one would believe that I actually accomplished what I said I accomplished. So, part one. A good procedure tells what the scientist did in steps that are specific enough so that another scientist can follow it. So that's one way we know we're going to have a good procedure. The next thing that we want to be able to do is that it's very clear what the independent variable is. Remember, the independent variable is what I change on purpose, and I'm going to change it at least 5 times. The idea here is that I want to change it enough times that I'm certain that there's some sort of pattern going on, but I don't want to change it so many times that it just takes a really, really, really long time to do my experiment. But the important thing is we want to see the trend, which is really like a pattern in our data. So 5 is kind of a good number because it's a pretty good amount for showing the pattern, but it doesn't take that long. Let me show you what I mean. So these are some of the patterns that we might be noticing. Does the dependent variable go up or down? Does it go up and down by the same amount, or does it go up more and more and more, or less and less and less? Those kinds of things. Let me show you an example. Maybe I wanted to know, is there a trend or a pattern between TV watching and test scores? So I did 2. I noticed that, yep, there's definitely a pattern. If you watch 0 hours of TV a week, you're going to get a 90% on your test grade. If you watch 15 hours, you're going to get 20%. Okay, well, that shows a pattern, but here's the thing. This just looks like I go down, 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 down. If I test more people than just 2, I notice that for a while, there's not much of a change. And then eventually it goes down more and more and more. So for a while, it doesn't matter. So if you're watching, a few hours of TV a week, that's okay. But noticing that the more and more that you watch, the lower and lower your test grade goes. So yes, this answered the question, but this gave me a much better answer. So changing my independent variable, which is how many hours of TV were watched, more than 5 times gave me a way better answer than if I just did it 2 times. So that's why we say that. So part 1 of a good procedure is that the scientist tells what they did. Part 2 is that I change the independent variable 5 times or more. The third part is that we test a wide of range as possible using the materials that we have. So what that means is, let's say that I wanted to do an experiment about how does the size of a frog affect how far the frog can jump. We had a tub of 10 different frogs. When I'm picking out my frogs that I want to use, the size of my frog is my independent variable. So I want to choose at least 5 frogs, right? But those 5 frogs that I choose, I don't want to choose 5 frogs that are about the same. I want to choose a frog that's maybe the smallest one in the bucket. And then maybe I find the one that's the biggest in the bucket. And then maybe I choose 3 more that are kind of a range in between. If I just pick the 5 smallest, I'm not really changing my independent variable 5 times. Or if I just pick the 5 biggest or the 5 middle. Because what again is I want to see that pattern. So the important thing is, did I test as wide a range as possible with the materials that I had? And sometimes that might be limited depending on the room size or the frogs that you can find here in Minnesota or those kinds of things. But whatever is available, I test as wide a range of the independent, the thing that I'm choosing on purpose. All right, so part one says that of a good procedure says that I tell other scientists what I did in a way that they can understand it. Step two says I take my independent variable and I choose 5 different ways to change it. And then part three says I change as wide as possible with the materials that I have. All right, what's the next thing? Well, the next thing says that I retest or repeat each data point multiple times. And usually 2 or 3 trials is a pretty good idea. Again, repeatability is important. So if whatever the trial that I'm doing is a little bit hard to count or a little bit hard to time or it's moving a lot, what I want to do is I want to make sure I'm certain of my answer. So I repeat it 2 or 3 times. Now, if my object isn't moving or I'm just counting something that's staying still, I might not need to repeat it multiple times. But if it's something, again, that I'm timing, so I have to start and stop my stopwatch, or if I have to count something that's moving, I want to do it 2 or 3 times. And then if I get similar answers 2 or 3 times, I can be pretty sure of my answer, and then I can find the average. So again, repeatability is important. If you're changing your independent variable, your dependent variable should be changing too. If you're not changing your independent variable, your dependent variable would also stay the same. So we retest the change in the independent variable to make sure that the results are the same or almost the same. If they are, we can move on. If not, we retest a 3rd time to make sure that we're certain of our answer, and then again we can find an average. So repeating each data point. So again, back to that frog example. So if I chose 5 different frogs, I probably would test each frog 2 or 3 times because there's going to be a lot of variety in how they jump, because maybe one time they jumped really far and then the second time they, their legs were tired so they didn't jump far as much. I can average those 2 trials to get my answer for how far did they actually jump. Then I take my new frog, and then again, 2 to 3 times do I let it jump. And then my 3rd frog, 2 to 3 times. So don't get number 3 of a good procedure and 4 of a good procedure mixed up. Part 3 was, and part 2 and 3, were that I changed my independent variable 5 times, like 5 different frogs. This part, good procedure number 4, says I repeat each of those frogs 2 or 3 times. And again, that only matters if it's some type of movement or change. If it's just counting something staying still or measuring something staying still, I probably don't have to repeat it 2 or 3 times. So something to kind of think about depending on what your procedure is. All right, the next thing is that we try to keep constant as many other things as possible. So now there might be things that we can't control, like weather patterns or time of day or those kinds of things, but we'll try to keep them constant if we can. So like in that bread example, we only want to change one variable, which is our independent variable, so that we can be sure of what's really going on. So with that frog example, the only thing I want to change is my size of frogs, kind of the breed of frog, or where they're jumping, or how I make them jump, or measuring in meters or centimeters. I want to keep all of that stuff the same, because otherwise I'm not going to be really sure what's causing those frogs to jump differently. So I want to make sure all the frogs are healthy, that they've eaten, maybe that they're wet enough so that they're not like dried out, because maybe that would affect how they jump. So keeping constant. So a good procedure has all 5 of those things. Let's review what they are. One thing is that I tell my scientists what I did so that they can repeat it. 2 is that I test my independent variable 5 different versions to see the whole pattern. Step 3 says that when I'm testing those independent variables, choosing them, I choose as wide a range as possible with the materials that are available. Number 4 says I retest each of those 2 to 3 times if I'm not certain of my answer. And then number 5 says I try to keep constant everything else. So those are my 5 parts of a good procedure. So when I give you a procedure, or I'm making you think of your procedure, those are the things you'll want to think about. So let's look at a procedure and let's evaluate and fill in some of that information. So this would be where you might want to be pausing the video and going back and forth to fill in these examples on your own handout while we go through them on this video. Okay, so here's the experiment. 10 seeds were planted in 5 different pots that contained 500 grams of potting soil. The pots were given the following amounts of distilled water, which is kind of like purified water, each day for 40 days. Pot 1 got 50 milliliters, pot 2 got 100 milliliters, pot 3 got 150 milliliters, pot 4 200 milliliters, pot 5 250 milliliters. So that is the experiment. That's what we were trying to figure out. All right. And then after 40 days, the height of each plant was measured at the end of the experiment. So I've got seeds in potting soil, in pots, and then they're getting different amounts of water, and at the end of the experiment, I'm measuring their height. So let's go through the steps of our procedure. So what is the problem that we're trying to solve? Now remember, the problem is a question. What is the effect of blank on blank? So think through what this was. So I was doing something with water and height and plants. So the problem that we're trying to solve is, what is the effect of the amount of water that I give a plant on the plant growth? So the amount of water is what I'm changing, and the plant growth is what I'm measuring. So the effect of blank on blank. All right. Then what is our hypothesis? Now remember, the hypothesis is if I change the independent variable, what will happen to my dependent variable? Because why do we think that? All right, so you think about that for a little bit. Go ahead and pause the video. Go ahead and try to think about what your hypothesis may be. Okay, well, let me give you one format of a hypothesis that would work. Maybe I say, if I water the plants with more water, that's my change of my independent variable, the plants will not grow as well. That is my change or prediction about the dependent variable, because when I get too much water in me, I just pee it out and get rid of it and not use it. So the plants would do the same with the extra water. So that is my hypothesis. If whatever I'm changing, then what will happen? Because why I think that, with either some type of personal experience or some type of scientific idea behind it. All right, so we have our problem, we made our prediction, let's keep going. The next thing we're going to want to think about is, what did I change on for purpose? Do you remember what it was? It was the amount of water. I had 5 different plants that I was giving different amounts of water to over the 40 days. And then the dependent variable, do you remember what this was? This was what was happening because I was giving different water. Well, this is your height of the plant after the 40 days. And then our next thing to think about is, what did we maybe try to keep constant? I always want you to give me at least 2. So let's think through this experiment. I'm changing the water, measuring the height of the plant. I want to keep everything else the same. So this is going to be things like, I'm going to use the same kind of pot that I put my seeds in. I'm going to give them the same amount of soil, the same type or brand of soil, the same amount of sun, the same number of days to grow. All of those things are going to help me be certain that if there's a difference in height, it's only because of the change of water, because everything else was constant, was the same. All right, let's keep going. So the next thing we might want to think about is, why maybe when the experimenter did this experiment, why did they choose 250 milliliters as the maximum amount of water to apply to all the plants? This gets to thinking of testing a wide of range as possible with my independent variable. Well, maybe they found out that if they put more than 250 milliliters, it would overflow the pots. And then if they used any more than that, it would not soak into the soil at all. So then they're not really watering the plant if it's just sitting in there. So this is this idea of testing as wide a range as possible. They found out that you really can't do more than 250 milliliters. So that's a good part of a procedure. The next thing we might want to think about is our data table. Now, we haven't really talked about this yet, so this is just going to be kind of your first example. So we think about what data we want to take. Well, we want to take our independent variable, which is how much water, and the growth, which is our dependent variable. So we set up a data table that kind of looks like this. The amount of water, that was our independent variable, goes down the side. So when we set up data tables and talk about them a little bit later, we'll always do that. Independent variable goes down the side. Here's my different amounts of water. And then the height after 40 days, I can't fill that in yet because it hasn't been 40 days. And then notice that I put the label with our numbers. This is going to be measured in milliliters. This will be measured in centimeters. And then after the 40 days, I can just fill it in. Now, you might notice that when I set up my data table, I only put one place for filling in height after 40 days. And so if you think about, well, what might be wrong with this procedure? Because remember, one of our 5 parts of a good procedure said to retest them 2 or 3 times. But think about this. So maybe that's something that you think, oh, wait, that's bad. We only tested the independent variable 1 time. But remember that the thing we're measuring, so why is that okay for this experiment? we're measuring plant height. There's no reason to get a different height each time. They're not moving. It's not timing something that's changing. We're just holding the plant and measuring the height. So remember that 2 to 3 trials is for things like when you have to start and stop a stopwatch, or you have to measure something that's moving, like the movement of a car or something like that. So remember that when we're thinking about procedure, there is a little bit of variety that would be okay there. All right, so that takes us through all the different little parts of our procedure, and then after this, we would maybe do a graph or make a conclusion, but at least this gets us thinking about the steps of our procedure. So let's review the 5 steps one more time. 1, I have the steps, I know what we did, so a scientist could repeat this experiment. 2, I tested my independent variable 5 different times with as wide a range as we possibly could. I didn't have to retest it for this experiment because the plants were not moving. We're not going to get a different, there's no reason to average our heights. And then the other thing is that we kept everything else constant. So this is a pretty good experiment. It's set up in a way that you'd be ready to actually do this experiment and see what happened. All right, so with that introduction, it's time for you to try some of this stuff on your own. So we just went through some notes. You have 2 more example problems on the handout, Scientific Procedures. So now what I want you to do is I want you to work through those examples, ask for help as needed, and then when you think that you're done, show Miss K before you submit that assignment. Thanks for listening. Go to work.